Hi everyone, we're the fMRIusic team, part of the Jahaniki Neurolab. Our project right now is preprocessing for analyzing neuroimaging modality of fMRI data associated with the emotional responses to music. Could you zoom in on the abstract? Okay. Functional magnetic resonance imaging, known as fMRI, is a non-invasive neuroimaging technique. It utilizes both signals to construct high-resolution images of brain activity from subjects instructed to perform tasks or respond to stimuli within an MRI machine. Extracting meaning from fMRI data requires choosing an appropriate raw dataset and performing numerous preprocessing steps to reduce noise and artifacts. This summer, the dataset accompanying the paper, Dynamic Intersubject Neurosynchronization Reflects Affective Responses to Sad Music, publicly available at openneuro.org, was chosen for analysis. This dataset contains scans of brain activity of 40 participants who were listening to music asserted to evoke either sadness or happiness. Analyzing this dataset will provide insight in how stimulus-driven changes in activity and connectivity in the brain correlate to emotional enjoyment and intensity. This term, the dataset was preprocessed through an fMRI prep pipeline, as well as using ICA-Aroma. In the fall, we will finalize all preprocessing and begin analysis with the aim to examine regions of the brain most connected to musical and emotional experiences.
Okay. So as mentioned before, functional magnetic resonance imaging is a readily available, non-invasive neuroimaging technique used to measure brain activity. Different signals and pulses produced within the magnetic field of an MRI machine are employed to observe relative activity in brain areas from sensory-related tasks or stimuli. Of these signals, as an MR signal, employed to indirectly detect levels of activity in specified brain regions using the blood oxygen level dependent or BOLD response. When neurons in the brain respond to a stimulus or task, they require oxygen from blood. Changes in MR signals result from blood oxygen levels heightening when there's an increase in brain activity, as opposed to a resting state. BOLD signals provide the basis for fMRI and allow researchers to compare resting and stimulated states of the brain. We have started pre-processing and analyzing data from an fMRI dataset. Pre-processing makes data easier to interpret and use. Pre-processing pipelines eliminate inconsistencies or noise in data, which can otherwise negatively affect a model's accuracy. The pre-processing step is crucial for ensuring the validity of our results. We use the fMRI prep pre-processing tool because it equips us with a high-quality, robust, easy to use and transparent pre-processing workflow. fMRI prep requires minimal user input while providing easily interpretable and comprehensive outputs. We have started pre-processing and analyzing data from our fMRI dataset, and we're looking to work on an analysis very soon. I think you should go to blog next, Aryan.
This summer, we published various neuroimaging blogs on our lab website, jneurolab.org/blogs. These neuroimaging blogs are tutorials on how to use various neuroimaging tools, written in detail and also in language that everyone can understand.
Our data is from a dataset used in the study about the dynamic intersubject neural synchronization reflects affective responses to sad music. In this study, there were 3 pieces that participants listened to, 1 longer piece deemed to evoke sadness, 1 shorter sad piece, and 1 happy piece. Participants were screened out after the trial, such that only participants that associated the appropriate emotion to the appropriate piece and had low familiarity with the piece were used in the analysis.
Firstly, the newly acquired data was preprocessed using neuroimaging tools such as fMRI prep and FSL. fMRI prep runs basic preprocessing steps. We then used FSL to run ICA-Aroma. ICA-Aroma is a method that identifies and removes any motion-related independent components from fMRI data. ICA, or independent component analysis, is a signal processing method to separate independent sources that coincide with each other. AROMA stands for the automatic removal of motion artifacts and removes ICA components from our fMRI data. This is an example of one of the fMRI commands that we ran on the ASDRP server through the use of publicly available Docker containers, which contribute to the reproducibility of our results. This is a visualization of one of the fMRI data from one of our subjects' runs. Here you can see slices of the brain as well as a 3D rendering. The brighter regions are areas with stronger bold signals, which means that there's more blood flowing to those regions and thus greater activity. Down below is a graph of the strength of a bold signal of a single pixel over time, which in this case is how active that portion of the brain was over the course of a piece of music.
This is our results for what we've currently done for this semester. We currently have the results from pre-processing with fMRI prep. One of the reasons fMRI prep is so useful as a neuropreprocessing tool is that it outputs a visual quality control, one HTML file per subject, in addition to the derivatives and the confounds, which are derivatives describing signals and noise that can be used to take denoising steps. Here are some of the visual quality control outputs of fMRI prep on one of our subjects. The first row of images consists of actual scans of the brain of a single subject, showing the different analysis run by fMRI prep for preprocessing the original scan. The images in a rough left to right order consist of white and gray matter segmentation, field maps that depict resting state activity, templates to correct structural image distortion, structural and functional activity overlays, and an image that highlights the signals in the region of interest as activity variability in the scan. And then the final image depicts the initial output and the final outputs after fMRI preprocessing from a distorted scan to a corrected scan. The graphs below show the different components that could be potentially unrelated to neural activity and thus removed later, potential time series where movement could have occurred that impacted the brain masks, and components with high correlation that could have further implications in the data and thus help us to further preprocess and remove variables that could impact our future analyses.
So firstly, the remaining data will be preprocessed using fMRI prep and ICA-Aroma. We then plan on implementing machine learning, and the first step is the time series extraction. The time series extraction feature is a preliminary step for machine learning on fMRI data that involves finding characteristics in the data that help solve a particular problem. Our next work in the fall will focus on preparing our data for machine learning.
This semester, as a team, we have gained knowledge in obtaining and working with neuroimaging datasets from past studies, as well as organizing the data into BIDS format. Along with successfully publishing blogs on a number of different neuroimaging tools, we made progress on preprocessing our data using fMRI prep, FSL, and CPAC, and will continue to preprocess and analyze the data for more solid conclusions. Overall, we hope our inquiry into this dataset will provide insight into how reactions to emotional stimuli differ in the brain. Understanding the underlying activity behind emotions has implications in research concerning those who respond to emotional stimuli differently, such as people who are neurodivergent and those with neurodegenerative diseases, and could potentially help with future therapy.
We'd like to thank Saharja and Ikia for our guidance and support throughout the research process. We'd also like to acknowledge the Aspiring Scholars Directed Research Program for providing us with the resources to conduct neuroimaging research. Finally, we would like to acknowledge Matthew Sachs, Asal Habibi, Antonio Damasio, and Jonas Kaplan from the University of Southern California's Brain and Creativity Institute, and Columbia's University Center for Science and Society for providing us with the data for preprocessing. You can scan the QR code in the resources to see them, and thank you for listening.